My name is Friedrich. I am finishing my third year here at Gladstone and UCSF in the bioinformatics program, and I'm interested in epigenetics, genomics, and bioinformatics. So I'm interested in understanding currently how you go from an embryonic stem cell to a more mature cardiomyocyte, so a heart cell. So how does a cell decide to go from a cell that can be anything it wants to be? It can be a brain cell, it can be a limb cell, it can be a heart cell. How does it decide it's going to be a heart cell? There's a lot of experimentalists here in the Gladstone that are really good at working on this sort of question in a wet bench sort of way. But I'm interested in looking at it at the dry lab sort of way, meaning I want to look at the data they produce, and I want to see if I can determine patterns that can help us understand how this process occurs. So I'm pretty lucky. I grew up in the South Bay here in California, and my parents were, both engineers. I've had a lot of people say, oh, in high school, like, you should, pursue this further. I just didn't really, I guess I didn't listen to them. It was that professor in college. He was a life science professor at UCLA, and he just believed in me, and I think that's a lot of what it takes to keep going in science. It's a lot of criticism, and you can potentially not take it too well. And so I think having that mentorship, having someone believe in you and put in the time and effort to back up that belief in you was really what kind of got me interested in pursuing it further. I'm really interested in specifically determining if there are certain preferences in the cell where a protein might bind DNA and cause it to maybe influence it to become another type of cell, which it might be a heart cell at a later point or something like that. So how does this process occur? How does DNA control this process of a cell becoming a heart cell from a type of cell that can become anything? Understanding how the cell works may sound totally abstract, but over time, the way the collaborative environment UCSF could help it maybe fine-tune experiments in such a way that it could be applied in some patient that needs some sort of cell replaced or some organ replaced or something like that. You could potentially see something where someone could take some cell in your body and make it into any other cell you want it to be. And let's say you have a stroke, and you need to replace those cells that were damaged. So we could potentially do that for you. They right now have, beating hearts in a petri dish. It's not totally impossible to maybe someday have a full functioning heart that, some patient may be able to have their heart replaced in some way. Over time, understanding how that single cell works and then asking how it works with other cells, and then how does this work in a 3D environment, not just in a plate level? How do these cells feed back in such a way that it creates an organ? That's ultimately would be a dream of mine to someday be able to look at it at that level. Yeah.